Okay, so I'm coming from the Institute of the Chemistry, Technology and Metallurgy, National Institute of the Republic of the Serbia, which is part of the University of the Belgrade. And I'm pleased today to present all the research that I have done with my colleagues here titled as Analysis and Characterization of the Novel Exopolysaccharide produced by a probiotic strain of Lactobacillus reuteri B2 and its microencapsulation using an alginate-based biomaterials. So firstly, I will speak you about, can you hear me?
Yes, we can hear you.
Okay, sorry. So firstly, I will speaking about generally about probiotics, prebiotics, and exopolysaccharides. So as already known, the World Health Organization defined these probiotics as live microorganisms which, when administered in adequate amounts, confer a health benefit upon the host. Mostly the lactic acid bacteria and bifidobacteria are the most common types of the microbes, and these are presented as probiotics, but also there can be found certain yeasts and bacilli. About the prebiotics, World Health Organization defined them as a non-viable food component that confer the health benefit on the host associated with modulation of the microbiota. Exopolysaccharides that are coming from the bacterial cells are the high molecular weight carbohydrate polymers often secreted by cells into the extracellular environment. So our research is divided in the three phases here, where you can see that the first phase we have done the probiotic assays of the strain that is isolated from the C57BL6 mice. Then in the second phase, we worked on the microencapsulation and material characterization. And in the third phase, we worked on the exopolysaccharide characterization. So about the probiotic assays, we have done firstly the isolation of the fecal bacteria, then a strain identification using 16S rRNA sequencing, which is confirmed with EPI tests, then acid resistance and bile salt tolerance, and antimicrobial activity of the Lactobacillus reuteri B2 strain. In the second phase, we worked on the material characterization and microencapsulation. Firstly, we have done the optimization of the probiotic encapsulation, enumeration of the viable cells, and survival of the free and the microencapsulated cells in simulated gastric juice. After, in the third phase, we worked on the exopolysaccharide characterization, where we have done isolation, extraction, and purification of the exopolysaccharides from the lactic acid bacteria from the Lactobacillus reuteri B2 cells. Then we analyzed them on the chemical and biological activity. We analyzed chemical analysis. We worked with, we have done with FTIR, NMR, HPLC, and SEM microscopy. While the biological analysis we have done using the MTT test, antioxidative activity, and adhesion of the Lactobacillus reuteri B2 strain to the HT29 cells to find the mechanisms of the probiotics. So in the characterization of the probiotics of the Lactobacillus reuteri B2 strain, firstly, we have done the strain identification using 16SR DNA sequencing with universal primers, which are the Uni16S forward and Uni16S reverse primers. That this strain was identified as a Lactobacillus reuteri B2, and the excess number of in the gen bank are presented here in the slide. So on the following tables here, you can see that we have done the acid resistance and bile salt tolerance of the Lactobacillus reuteri B2. And we can see that on the very low level of the pH at 2, we obtained the viability of the cells, which is more than 64%, which is very good result. This result is confirmed using the following experiment of the bile salt tolerance, where we obtained that the viability of the cells was around the 37%, in modified MRS with oxagal of the 0.30 percentages. After this experiment, we have done also the antibiotic susceptibility of the Lactobacillus reuteri B2 strain using the 5 different antibiotics. And here, as you can see, the resistance, this strain is a resistance on the vancomycin, which is actually expected because this one antibiotic is the negative control for this experiment. And intermediate sensitivity we obtain against versus amixocillin and tetracycline, while the high sensitivity we obtained against the ampicillin and cephalotin. On the next slide, you can see that we have done also the antimicrobial activity using disk diffusion method against the 9 different pathogen microorganisms. And also here I would like to say that the highest antimicrobial activity Lactobacillus reuteri B2 strain obtained against Klebsiella oxytocin J7, then Shigella flexneri, and Staphylococcus aureus. Here I would like to mention that Lactobacillus reuteri is obtained the highest antimicrobial activity against the Klebsiella oxytocin J7, which is actually extracted and isolated from the same source as the Lactobacillus reuteri B2. After the probiotic tests, we asked which material should be used for encapsulation of the probiotic cells of the Lactobacillus reuteri B2 in the simulated gastric juice. And we have done the characterization of the material for encapsulation of the Lactobacillus reuteri B2 with optimization of the probiotic encapsulation, where we have done the encapsulation yield using the following formula here, where the N presented the entrapped cells into the microencapsulation beads, while the N0 presented the free cells added into the biomaterial mixture during the process of the microencapsulation. On the left part of this site in the contour diagram, you can see that the encapsulation yield of the starch malate were around the 70.2 percentages, while the encapsulation yield of the sodium alginate was around 49 percentages. Then we have done the starch malate characterization presented here in the set of the experiments, where we have done the degree of the sensitivity, viscosity, reaction activity, then swallowing capacity, and the moisturizing absorption of the pure starch and the starch malate. And in the table here presented, you can see that we obtained the better results with the starch malate than with the starch, the pure starch. After that, we have done the experiment of the encapsulation yield and viability of the Lactobacillus reuteri B2 in 2 different materials, the sodium alginate and sodium alginate with starch malate beads. After experiment, we obtained that the encapsulation yield of the cells, the encapsulation yield was 88.4 percentages for the material with starch malate, while the 70.2 percentages was the encapsulation yield only with sodium alginate and probiotic. Also, the viability that we obtained in this experiment were higher in the material with the starch malate, which was presented as 7.88, while the viability of the cell form, viability of the Lactobacillus reuteri B2 strains from the beads of the sodium alginate was 7.80. Then we have done also the analysis of this material using the SEM analysis, TGA, DTA, and TIR analysis, where we, after the modification of the starch with malate acid and sodium alginate, we obtained the specifically peaks of the material that confirmed this structure. And also in the pictures of the cell microscopy, you can see that we obtained actually two different materials with the higher number of the Lactobacillus reuteri B2 cells in the material with the starch malate. After this characterization, we have done also the experiment in the simulated gastric juice, which was during 2 hours, where we would like to see the survival of the Lactobacillus reuteri B2 cells in the free form and in the form of the microcapsules in these two different materials. The results that we obtained after the experiment, here you can see that the highest viability of the cells of the Lactobacillus reuteri B2 cells were with the material with starch malate, while the less viability was in the material of the sodium alginate. Also, the free cells were obtained less viability than the cells that were encapsulated. After the probiotic characterization and microencapsulation, we entered in the third phase of our research, where we would like to see how these exopolysaccharides that are secreted from the Lactobacillus reuteri B2 cells obtained characteristic and are identified from the chemical and the biological activity. So the exopolysaccharides are generally produced from the microbes, mostly of the lactic acid bacteria and bifidobacteria, and this is very important for the biochemical industry and the pharmaceutical researchers because of their possibility to produce the exopolysaccharide, which are involved in the beneficial properties for the human health. So firstly, we have done extraction and the purification of the exopolysaccharides from the Lactobacillus reuteri B2, where we inoculated these species into the modified MRS with 10 percentages of the saccharose, and after overnight incubation, we added the 80 percentages of the TCA or trichloric acetic acid to degrade the proteins inside the system. And after the fermentation and the centrifugation at 8000 G, we obtained the supernatant that where we used the ethanol participation of which were two times of the which presented in the two times of the volume from the supernatant. And after the incubation from the 24 to 48 hours at the 4 degrees, we obtained the raw exopolysaccharide pellets. After rewashing this pellet with the deionized water, and after vortexing, we obtained the exopolysaccharides, which were then further go to the dialysis from the 10 to 12 kilodaltons, and after that we obtained the purified exopolysaccharides. So these purified exopolysaccharides then we sent to the lyophilization for easier identification in the chemical and the biological properties. About the chemical structural characterization of the exopolysaccharides, here we have done the HPLC, NMR, FTIR, and SEM microscope analysis, and based on the NMR analysis of these exopolysaccharides, we obtained that mostly we have alpha D-glucan and beta fructofuranose. Multiple signals in the NMR spectra showed that the glucan contain alpha 1,4 and alpha 1,6 linked alpha glucopyranose residues. Also, we obtained a very weak signal of the hydroxyl group in the spectra, indicated of the possibility interaction this group with deuterium in the deuterium water solvent converts to the OD groups. The NMR chemical shift data, together with the FTIR data and HPLC techniques, improved approach to the structural classification of the exopolysaccharides. And on the right part of this slide here, you can see the FTIR spectra, actually that obtained that present the ticks which are connected with the structure of the exopolysaccharides. Then, after this analysis, we have also done the characterization of the exopolysaccharides from the Lactobacillus reuteri B2 by SEM microscopy. And here you can see that we obtained these pictures on the three different magnification, 10 times, 30, and 50 times. After the chemical analysis, we have also done the biological activity of the exopolysaccharides. Firstly, we have done then antioxidative properties from the exopolysaccharides of the Lactobacillus reuteri B2 using the ABTS test, and we are calculating the scavenging activity using this formula here. And from the graph presented on the left part of the presentation, this slide, you can see that while we are increasing the concentration of the exopolysaccharides from the Lactobacillus reuteri B2, the percentages of ABTS scavenging ability is also increasing. We compared these results with the ascorbic acid or vitamin C. Then the biological properties of exopolysaccharides from Lactobacillus reuteri B2 was also tested using the MTT test, where the cell viability of the human lung fibroblast cell line of MRC5 treated with different concentration of the exopolysaccharide from the Lactobacillus reuteri B2, 0.5, 1, and 2 milligram per milliliter for 24 hours. Determined by MTT assay. The results that we obtained here showed that the analyzed concentration of the Lactobacillus reuteri B2 of the exopolysaccharide were not non-toxic to MRC5 cell line after 24 hours of the treatment. Also, we have done the adhesion activity of the Lactobacillus reuteri strain on HT29 human intestinal cell lines, because here we would like to see how the mechanism of the probiotic it works, and the adhesion ability of the Lactobacillus reuteri B2 was tested on the HT29 cell lines, and the percentages of adhered cells comparing to the edit cells was 1.18 percentages, which presented a very, very good result. So about the future researches, the future researches will divide it in the three parts. Firstly, we would like to do some modifications of the functional groups of the exopolysaccharides for finding the novel biomaterials for microencapsulation of the probiotic cells, as well as the novel carriers, also for probiotics and drug delivery in the camp of biomedical science. The functional attributes also of the polymer or its producing strains need further validation under in vivo condition, and development of the novel of new equipment that could be used in the large commercial processes, both in the food industry and in the pharmaceutical processing industry, presents the challenge for researchers in this field, so as well as related researches into accessing the efficiency of microencapsulation of the gastrointestinal tract using an animal model. As conclusion here, we can say that Lactobacillus reuteri B2 strain is a novel probiotic strain isolated from the mice species of the C57BL6 according to the results obtained from the probiotic assay. The microencapsulation of the Lactobacillus reuteri B2 obtained the better viability of the probiotic strain with sodium alginate and the starch malate biomaterials, and exopolysaccharide extracted from the Lactobacillus reuteri B2 strain showed that the chemical and biological activity were evaluated to determine its potential health benefit for the host. Now I would like to acknowledgement for this work. This work has been supported by Ministry of the Education, Science and Technological Development of the Republic of the Serbia. We have obtained 3 grants from the My Institute, the Institute of Torlak, and the University of the Belgrade, the Department of the Organic Chemistry. And special thanks to Katarina Novovic, PhD from the Institute of Molecular Genetics and Genetic Engineering. Thank you very much for your attention, and now I am able to answer on your questions. And also here on the bottom of the slide, you have my mail address, so also the questions can be sent in this address.